Hello, my name is Erin Newbern and I lead the Field Application Scientist group at Personalis. In this video, I'll be discussing our ImmunoID NeXT platform, a multidimensional tumor immunogenomics platform that can enable investigators to obtain comprehensive biology from limited and difficult samples. So first, just a bit about our company, Personalis. We were founded out of Stanford University in 2011 with a mission to really transform the development of next-generation therapies by delivering comprehensive molecular data about each patient's cancer and immune response. We are located in Menlo Park, California, and we are a CLIA certified and CAP accredited laboratory. Our two key applications of our platform are for immuno-oncology biomarker discovery and also for neoantigen prediction for personalized cancer therapy development. Currently, immunotherapies are rapidly having a major impact on patient treatment. While combination therapies seem to be the best strategy, as we design more rational combination approaches, it's also becoming more important to understand the mechanisms of hot and cold immune tumors and the complexities of the cancer ecosystem. This figure shown here was taken from an interesting review published earlier this year in Nature. And the authors have put forth this tumor immune classification cycle and how it can be used to direct cancer therapy, basically by classifying tumors into subtypes according to their T cell presence and T cell distribution. And also, we're really seeing many ambitious approaches and progress being made underway in immunotherapy clinical trials. However, we're still faced with many obstacles. First of all, there's unfortunately many patients that are not responding to these treatments or combinations of treatments. There's also very heterogeneous tumor resistance mechanisms out there, and also, importantly, a lack of solid biomarkers in immunotherapy. For immunotherapy biomarker discovery, PD-L1 expression is not a perfect metric for determining patient response. While it does provide some clinical utility, most are going beyond PD-L1. Another metric in clinical practice is microsatellite instability status. Likewise, mutational burden is an additional emerging metric suggested for determining patient response, yet does not entirely delineate the responders and the non-responders in all cancer types. So there are many further biomarkers for immunotherapies currently under investigation, as you see listed here on the slide, that are becoming a next generation of potential predictors to hopefully guide patient treatment. It's also thought that a single metric may not be entirely predictive for immunotherapies, but a more complex biomarker may be needed. And as we take on the obstacles of immunotherapy biomarker discovery, we know that these are very critical, precious patient samples that have been acquired. Traditional processes often require multiple samples to be sent to multiple vendors to get the essential data needed in the study. So for example, perhaps you may send a blood sample out for HLA testing, another tissue section for expression analysis, and then send another tissue section for assessing mutational burden. And the result is that sometimes investigators must make difficult decisions. There may be trade-offs about which particular assays can be ran when there is limited sample, or just dealing with complex data integration after working with several different platforms. So ImmunoID NeXT was really purpose-built for immuno-oncology biomarker discovery and designed as a solution to use a single sample preparation and to consolidate multiple biomarker assays into one. So we've designed our approach, ImmunoID NeXT, as a one-stop solution that enables multiple analytics to really maximize data generation. And this is the first platform to enable comprehensive analysis of both the tumor as well as the tumor microenvironment. It enables investigation into critical areas such as detecting neoantigens, looking at tumor escape mechanisms, HLA typing, tumor mutational burden assessment, and gene expression. However, now we've added a deeper view, more complete picture with this latest design, with the ability to interrogate the T cell repertoire composition, microsatellite instability reporting, as well as oncoviral detection and immunocellular deconvolution. And this is all from a single sample prep. So this really makes this assay very innovative, really the first of its kind approach. Undoubtedly, there are many obstacles still facing investigators in biomarker discovery. An initial obstacle that we'll focus on here is the ability to work with more difficult sample starting materials like FFPE in your DNA and RNA sequencing process. How do you maximize data generation from these specimens that may be either more limited or more difficult to work with? In our lab at Personalis, we receive a variety of different sample formats: FFPE, whole blood, fresh frozen tissues, PBMCs, cell lysates. However, FFPE tends to be the most predominant. And shown here on the left is a snapshot of several FFP materials that have recently came to our lab. And as you can see, these materials can come in a variety of different qualities and quantities. And this is frankly the reality of working with clinical FFPE materials. But at Personalis, we've specifically optimized to work with these more challenging sample types and have currently a greater than 90% success rate for any sample format we receive in our lab. The data here on the right shows the percentage of samples passing our DNA and RNA extraction QC criteria. These samples come from many different biomarker and personalized cancer therapy customers and many different sample collection sites. And you can see we have excellent performance on both fresh as well as archival FFPE materials. And we also perform this in a sample-sparing way. So we do a dual extraction of both DNA and RNA from the tumor specimen and can work with as little as 50 nanograms for DNA and as little as 100 nanograms for RNA in our optimized process. ImmunoID NeXT is really powered by the unique, very innovative assay design. First, by the depth of sequencing coverage and also the proprietary augmentation that we perform. We perform an average 300x coverage across the entire whole exome footprint, but this is an enhanced augmented coverage. ImmunoID NeXT utilizes our proprietary ACE technology to provide augmented coverage of difficult to sequence genomic regions across the entire whole exome sequencing footprint, so more uniform coverage throughout. And I'll explain this more in the next slide. And this is also paired with our optimized whole transcriptome sequencing of the tumor specimen as well. Secondly, we also provide boosts of ultra-deep coverage in critical areas of importance for oncology, with clinical-grade sequencing coverage across cancer driver genes for both solid tumors as well as hematological malignancies, and also ultra-deep coverage of the TCR and BCR gene regions. Some additional benefits of our very unique assay design is that we also have enhanced specific targeting of other loci, including HLA class 1 and class 2 loci, MSI-related loci, and also the ability to pick up oncoviral detection. And as mentioned, our ACE chemistry forms the foundation for our assays. Unfortunately, the issue with the standard conventional off-the-shelf sequencing assays is that these approaches have gaps and inconsistencies in sequencing coverage. And this is what we've recognized and addressed with our ACE approach. So for example, this is demonstrated here on this schematic on the left. So in blue, this is representing a gene using a standard exome sequencing run. And if we look across the gene-wide, you have what I call peaks and valleys in sequencing coverage. Areas sufficiently covered, but others that are also suboptimal. And even if sequenced at high depth, these approaches, the standard conventional approaches, fail to cover whole exonic regions. And this is shown here by the red dot. And if variants reside in these regions, would be undetected and would be completely missed using this standard approach. And if these areas harbor a variant that results in an immunogenic epitope, for example, it would be missed by the conventional assays. And this is what we have recognized and addressed with our proprietary ACE. So essentially what our customers receive from us is basically the blue plus the green to result in more uniform coverage throughout the genomic footprint. And this leads to ultimately a more accurate assessment of neoantigens, but also a more accurate assessment of the mutational landscape and biomarkers. So to summarize, ImmunoID NeXT is designed to be a universal genomics platform for immuno-oncology. The NeXT platform combines highly sensitive exome and transcriptome sequencing with advanced analytics and best-in-class data delivery. We are consolidating multiple biomarker assays into a singular approach utilizing one sample prep, and with the needed scalability. This assay is performed in our CLIA CAP laboratory on NovaSeq instrumentation, and also with the reliable support of both our project management and field application scientist team. So we are very excited with our extensive capabilities to provide a complete look at the cancer ecosystem. If you'd like to hear more regarding our ImmunoID NeXT multidimensional assay for immuno-oncology biomarker discovery, please contact us at info@personalis.com.